Eugenio, you already talked about the advantages of three-dimensional cell models. And you also mentioned that this is a well-established technology, but somehow these cell models have become a prominent focus over the last few months. How can the 3D models be used in drug discovery? And what are the challenges when using them, especially when you have to deal with miniaturized and fully automated applications?
Yeah, it is established technology, in my opinion, especially in oncology, has been extensively used. I mean, of course, there is an advantage is to have a 3D model that will resemble more physiologically a tumor, for example, rather than having a 2D layers on a plastic that is not exactly the most physiological model that you can have. However, in my opinion, any model has limitation and any model has advantages. 3D give this complexity of the cell-cell interaction in a more structured way, but it brings also complication for analysis, for example, very complex data sets to analyze with microscopy, for example. It brings also relatively complex physiological limitation. For example, there is no vascularization normally in a 3D model, so that means you get a very hypoxic core of the organoids, and this complicated the life. But still, what is good in a 3D model or a 2D plus model, what I say is that you can do co-culture. You can put a different system together. And this, of course, is benefit because then you start to get really close to the physiology, to what cells interact. I mean, things about the brain, if you use only neurons, is not a brain. A brain is much more complex. There is astrocytes, there is microglia, there are oligodendrocytes. So rebuilding a 3D structure is, in this case, an advantage. We have, for example, developed recently a blood-brain barrier model based on human-derived cells from stem cells that is resembling very much the physiological condition of the human brain. And that's also a model that is very important. I mean, we see now, for example, that there is the treatment for Alzheimer's disease with antibody that is becoming a reality. But we do know also that this treatment are caveats. For example, 1/3 of the patients develop edema. That means the blood-brain barrier has a play in this one. And now we can predict now, for example, can we use this model to predict which antibodies in which patients treat edema and what we can do to avoid that? Because, I mean, of course, if you want to treat a patient and you get the brain edema, is not exactly what you want. There are the advantages to using 3D models, so that's for sure. They get closer to the physiology. That's one of the advantages, in my opinion.
Do you see the use of these 3D models mainly in the early drug discovery process or better in later stages of the drug development processes?
My opinion is more in the late stage, yes, where you can really model more complex system to predict eventually adverse effects that are never good when you go to clinical trial, and also to predict efficacy. So that's I think where the 3D model, in my opinion, was promising. Thought there has been recently, for example, a publication where 3D model organoids reimplanted in a rat brain have been shown to reintegrate within the brain, so to innervate and vascularize and get integrated in there. So 3D models of organoids can eventually become interesting also as a real medical application, so for regenerative medicine. And that's still at the beginning, but it will develop more and more, in my opinion, as well.